I'm Jill Woodward. We're at the NIH in Bethesda, Maryland, and I'm here with Dr. Marston Linehan. Dr. Linehan, tell us what you do and tell us about the urologic oncology branch.
Right. At the urologic oncology branch, we study both the clinical and laboratory basic aspects of Birt-Hogg-Dubé syndrome. We started working on this in the early 90s. We see patients with kidney cancer that runs in families with hereditary kidney cancers. We have a knockout mice model with a very robust laboratory program, but our real model is the human model of cancer. And our goal, of course, is by studying the BHD pathway, both in the laboratory and in our animal models, to develop a therapy for BHD so that one day we don't have to recommend surgery to these patients. So our clinical approach is to recommend surgery when the largest BHD kidney tumor reaches 3 centimeters, that size. Now, we have not yet had a patient develop metastasis, have the cancer spread when patients are managed in that fashion. Now, we've had a number of patients, sadly, who developed metastatic cancer and went on to die with BHD kidney cancer, sadly. Their cancers either weren't detected and they got very large and they spread, or they were very large before we saw them and had already spread. So we're very encouraged about that. We're very encouraged. Now, these patients, we've switched now to managing many of these patients with, instead of doing an open operation, doing them with a robot, doing robotic surgery. We've always felt that if we could do these surgeries minimally invasively, in other words, with less incisions, that would be a benefit to these patients. So in many cases, we're able to do that.
Dr. Linehan, talk about the process of getting a drug from the lab to the bedside. How feasible is that?
I think getting from the gene to a drug is very feasible. Although I'm humbled by the complexity of that journey. Every cancer gene is different. And every cancer gene we're going to have to fight one at a time, including folliculin, the BHD gene. We, and I say we, meaning the field, know a lot and are learning a lot about this gene and this pathway. We already have some very clear directions on potential therapeutic approaches. The way you identify drugs is by looking at these pathways and predicting what might affect them. You test these, and we made a kidney cancer cell line from one of our patients with BHD, so we can grow that kidney cancer in experimental animals and see how drugs work there. We can look at these drugs in the laboratory. We can also look at our transgenic models to see how this class of drugs or this drug will work. I have held off so far on doing a clinical trial because it's just judgment, because I want to go with the best possible drug. I don't want to delay the work by doing a drug that's only part way. So it's, it's just a judgment call. We're getting close to the point of starting a trial. I would be surprised if we did not initiate a trial, a systemic therapy of patients with BHD kidney cancer, either later this year or early next year, spring of 2012. We will see. We're looking at a number of candidates right now. We're very encouraged about it, but it's going to be a long process.
Can you talk about the other forms of hereditary kidney cancer and how that affects our understanding of BHD and vice versa?
We study 7 different types of kidney cancer, of hereditary kidney cancer. We study von Hippel-Lindau, VHL, a hereditary type of clear cell kidney cancer. We study hereditary papillary renal carcinoma, which is a very rare type of type 1 papillary kidney cancer. Birt-Hogg-Dubé, of course. we study another less rare type of inherited kidney cancer called hereditary leiomyomatosis renal cancer, or HLRCC. And we study families with, recently we've expanded our study of families with something called succinate dehydrogenase kidney cancer. Now each of the genes and the pathways of those genes that cause these different types of inherited kidney cancer are different from each other. However, we have found that they all have in common that each one of these pathways is fundamentally a metabolic pathway. So we have learned a lot about BHD, the folliculin gene pathway, from studying these other pathways, and we've learned a lot about them by studying BHD. It was BHD that originally got us in to understanding that kidney cancer is fundamentally a metabolic disease. Now, when you think about it, targeting a metabolic disease is very, very different than the traditional way we think about treating cancer. So I'm very hopeful that understanding this fundamental process is going to give us approaches to therapy that we never would have dreamed of.
Doctors who treat BHT seem to agree that apart from avoiding smoking, there aren't very many lifestyle factors that a BHD patient can do themselves to avoid getting cancer. However, I recently read a book by T. Colin Campbell called The China Study, in which he suggests that diet may play a role in tumor growth. What's your opinion on that?
I'm not aware of that study exactly that you're referring to, but the more I study these cancer genes, the more I study these cancers and these pathways, I think diet and exercise are incredibly important. I think that in some ways, exercise may be the best drug for cancer ever developed. The more we study what that does, how energy consumption affects these cancer pathways, I wouldn't be surprised at all if diet didn't play a huge factor in many cancers, and also kidney cancer. If you look at all the cancers in the body, and you look at a relationship between body mass index, how heavy people are, and their incidence of cancer, kidney cancer is among the highest. There's a very clear correlation between body mass index, overweight, and incidence of kidney cancer. So it wouldn't surprise me at all if that wouldn't be seen also in the families. We've not studied that specifically, although it's just a matter of getting the people to do it. I mean, getting enough people to do it. We have the data for that. We're hoping to look at that this year. But I think that's an incredibly important, I wouldn't say underappreciated, because a lot of scientists know this is important, but the more we study it in all of our different kidney cancer genes, it just looks like one of the most central part of this cancer pathway, energy consumption.
How will new technology affect kidney cancer research?
It took us nearly 7 years to find the BHD gene. We did it, you might say now, the old-fashioned way. We got all these families, we drew their blood, extracted their DNA, ran genetic linkage analysis to trace the gene in the families. Today, you could do that work with whole genome sequencing all the genes in the cell, in the body, and I'm sure do that much more quickly. Well, it's the same thing with understanding the fundamental aspects of these cancers. In other words, we're using all sorts of new technology with mass spectroscopy, liquid chromatography, in some cases whole genome sequencing, in some cases whole genome array studies, whole genome array studies of microRNAs, and all sorts of new technologies. And those type techniques can just make things go quicker, give you new insights you wouldn't have known before. Whereas in the old days, which wasn't that many years ago, we used to look at expression of one gene at a time to see if a certain gene was turned on, or maybe one protein at a time. Now we can look at 37,000 genes at a time. So you can look at whole clusters of genes. Many times the problem you get into is you get so much information, sometimes it's a challenge to interpret it all. But that can help enormously.
Based on your experience in learning how to do research, what advice would you give an early stage researcher?
I think that for a young person coming into the field, I can't think of anything more exciting to do. This is a very big question, cancer, and we have a long way to go. But we know a lot, and there are a lot of tools to use to study. I was meeting with one of the leaders of NIH. We were just talking about things, and he said to me, "Marston, you have been doing translational research 25 years before the term was even invented." By that he meant translational research means doing things that relate to patients, using patients as a model and going to the laboratory. I said to him, that's the only model I know. It's the only model I've ever done, is studying the human model of cancer. So I would say for a young investigator coming into the field, for him or her, if they can, get into an environment where what they're studying is very translational to the human model, to humans with the cancer, that it not only makes it more exciting, but it's not just that. You learn an enormous amount from patients. If you're working in this area, just try and do the best science you can and try and relate it as much as you can to the patients with the cancer. Because otherwise, if you're not careful, you can spend your entire life studying something that doesn't, it might be an important thing, but it doesn't relate to human cancer. And we need those people. We need them working on to help these patients.
Dr. Linehan, thank you very much for your time today.
Thank you. I enjoyed it.